for like the rest of the unit. And actually kinases we talk about for the rest of the semester. So if you don't know what those are, this is a good chance to tell you, hey, these are really important. They're going to be on your exam. We're going to cover it for like a long time. So please make sure you know this. So if you read these points on the study guide and you have an idea of specific concepts that for sure will be on the exam, hopefully it piques your interest when we actually come across that slide during class. Rather than feeling like, is this slide important? Is this slide important? Because you see these keywords and these sentences and how these concepts or questions are asked, when it comes up in lecture, hopefully you'll be able to identify, oh, that was actually a concept that was asked in the study guide. And if you look at the study guide, you'll see like, these, some of them are very rudimentary, like, okay, what are enzymes, what are kinases, like it's a very definition type of thing, right? But some of them are like, okay, how does creatine supplementation improve resistance training adaptations, right? That actually relates to phosphocreatine resynthesis and how it's regenerated during resting periods, okay? As we'll cover today, essentially creatine allows you to do more work, and more work results in more adaptation, okay? So ultimately, you could answer these study guide questions as like a one sentence and done, but realistically, a better way to answer these would be to look at all the ways that this concept is talked about in that lecture, and to be honest, in several lectures, and tie them in together, okay? There's a very simple, like 1 or 2 sentence way to answer some of these, and then there's like a fairly long, drawn out, more thorough way to answer. So for example, if we go down to hormones, how does epinephrine influence muscle enzymes? Be specific. And how does it change? And how does it change in those enzymes influence fuel mobilization? Be specific. Okay. So what we'll learn about a little later in the semester is that exercise intensity influences the epinephrine response, right? Epinephrine is your fight or flight response. So the harder the exercise, the greater the epinephrine response. What we'll also learn is that the release of epinephrine interacts with specific enzymes in the muscle that will help mobilize glycogen use or mobilize fat use, right? And it will accelerate those different metabolic pathways so we can use those different fuels for the specific types of exercise we're using or we're performing, right? So yes, you can just say exercise increases epinephrine response and it mobilizes substrates. That's a very simple answer. But as we'll learn later in the semester, you're going to have to name the specific enzyme that each activated by epinephrine. Hormone-sensitive lipase, glycogen phosphorylase. How do they do that? It increases phosphorylation of glycogen phosphorylase, glycogen breakdown increases, more substrates used. And that's specifically during high intensity exercise. Low intensity, it activates hormone-sensitive lipase. Lipolysis goes up. More free fatty acids, those go to beta oxidation, those supply more ATP for aerobic exercise, right? So notice how, like, I said the same thing, but one was very general and generic. The other was much more specific, applying to specific types of exercise and which specific substrates and fuels. Does that make sense? So, yes, we cover those enzymes in the hormone lecture here, but also we're going to go over glycogen metabolism, where we'll talk about glycogen phosphorylase, right? So yes, glycogen phosphorylase comes up again in relevance to hormones and epinephrine, but we're going to talk about glycogen phosphorylase in this lecture and next lecture relevant to glycogen breakdown, right? So make sure we're understanding these very specific enzymes because we end up talking about them quite a bit in each section. Does that make sense? All right, so not that you have to know the answers to these points when you read the study guide, you absolutely don't. I don't expect you to. I just want you to be aware of these are the types of concepts that will be on the exam, and that when it comes up in the PowerPoint, you should take note that these are how it's worded, right? This is the concept that we'll deal with, okay? Now I'll also say, as a note, some students have said to me, oh, the study guide didn't cover this question, and this question, and this question, and this question on the exam. Well, no, I'm not going to give you a study guide that line by line goes through every single, like, one study guide question for one question on the exam. I'm not going to do that. Otherwise, I'm just giving you the exam. what you'll see is that all of these concepts that are on the study guide are asked multiple different ways on the exam, okay? So these are broad arching topics, and the more thoroughly you answer it, the more likely you're gonna get better understanding of that topic as a whole and be able to answer any type of question how it's asked on that topic on the exam. Does that make sense? All right, cool. So just throwing that at you. All right, so we are gonna, gosh, this room is like, Tuesday I was sweating, it was hot in here, and then I was cold, it's freezing in my office. I'm like, face is freezing, body is warm now, it's so weird. This room I can never get to breathe. Okay, so we're gonna start off talking about enzymes and the phosphagen system. Okay, so largely these first few lectures on bioenergetics, they are relatively reviewed from 346, but obviously there's going to be points that we're talking about metabolism just a little bit more specifically than we did 346. So yes, there will be new terms, new proteins, new enzymes thrown at you, but who are the major players we've already learned in 346? Well, there's glycogen, there's PFK, there's pyruvate, there's NAD and NADH. We've already heard about these things before, right? So those are the key players, but now we're going to talk a little more specifically how all those regulate metabolism. So just keep that in mind. So if we look at what goes on in the cell, obviously we have the mitochondria that are, we all remember from Nitrogen Bio, the powerhouse of the cell, right? We have the mitochondria that have very specific locations in the cell. We're learning more and more about molecular biology relevant to exercise every day, considering that now science is advancing more in our field in that direction. So now we realize certain mitochondria have more of a function for supplying ATP for sodium potassium pumping at the cell membrane versus mitochondria that are more closely located next to fat and lipid droplets right next to the sarcomere that can be used for energy, right? Same thing with like mitochondria located closely to the sarcoplasmic reticulum to supply ATP for calcium shuttling and calcium handling versus mitochondria located elsewhere that might be more specifically developed to break down and metabolize glycogen more aerobically, right? So we're learning about different aspects of muscle biology relevant to exercise every single day. All right, so a lot of what we're going to cover in this class is biochemistry, but we always try to take these biochemistry concepts and see how they apply to real-world training and application and adaptation. All right, so we'll talk a little bit about the nuclei, right? And the nuclei, this is where we have transcription and translation. Okay, well, we transcribe an RNA, and that RNA is produced, and then it goes through a ribosome that produces a protein, right? That is the process of protein synthesis. We have our myofibrillar proteins, those make up the sarcomere that produce force, okay? So these are all familiar components. Notice how the muscle fiber is highly organized, okay? But keep in mind, inside this cylindrical cell, there's fluid, right? Obviously, you can see it's compartmentalized where here's the sarcomeres, the myofibrils are structured in a very organized linear way, but there's fluids and water all mixing around, molecules, proteins bumping into each other. Sometimes contraction forces molecules in together. Sometimes enzymes being turned on literally draws two different substrates together to make sure an interaction happens, right? And all this happens because of contraction, exercise, and metabolism, okay? And these responses allow us to exercise and provide substrates and deal with the exercise stress more efficiently. Our body perceives exercise as stress, right? Right now, maybe you're stressed out because you're learning new stuff, right? From a mental standpoint, you're at rest, right? But as soon as you start to increase your energy expenditure, right? And what do I mean by increasing your energy expenditure? How can you quantify increasing energy expenditure? If the intensity of your work goes up, right? But why does work go up? What do you do? What do you have to do? Your heart rate goes up. Your heart rate goes up because
you need oxygen delivered.
More oxygen delivered to the muscle. What does the muscle do with the oxygen? Makes ATP, right? So if we go back to the first part of that question, our heart rate goes up to deliver more oxygen, to make more ATP. Why do we need to make more ATP? Because when we perform more work, we're using more ATP, right? So the increase in energy expenditure means that we are expending more energy. What energy does our cells use to produce work? ATP, right? So harder exercise, more intense exercise, utilizes more ATP. Low intensity but long duration exercise also uses more ATP, right? It just takes you longer to use it, okay? So when we look at substrates, okay, substrates and enzymes, okay, this blue molecule here, this is an enzyme. Here's a substrate, okay? A substrate is a molecule that fits in and works and interacts with an enzyme, specifically in its active site, okay? Enzymes, their active site is shaped or formed in a way that works with a specific molecule, a specific substrate, okay? Now, very much like Legos, you could have, or you have like shapes, you try to put a, square peg into a circle hole, right? It doesn't quite fit, right? But sometimes it might have like maybe an oval type thing that might kind of jiggle around and move into that circular hole, right? Sometimes you have other types of substrates and molecules that have a similar structure and can kind of fit into the active site, but not really tightly, right? So it might modify the enzyme in some way. But realistically, each enzyme works with a specific substrate to cause a specific reaction to occur, okay? And once you have that substrate interacting with the enzyme in that active site, we now have an enzyme-substrate complex, okay? Yes?
If the enzyme denatures, can it different substrate bonds with it?
It could, right? We'll talk a little bit about denaturing of enzymes in a little bit, but yes, we'll get there. Yeah. So one thing I want you to see here too is if we look at how the active site has changed, notice how when the substrate has now locked into the active site, what happened to kind of these two portions of the enzymes? They kind of closed, right? The structure changed, okay? So when an enzyme is activated or interacts with its correct substrate, the structure of that enzyme changes, right? And so if the structure changes, then the function changes, right? Maybe it's in some structure that is not able to do anything, but if the structure changes into the optimal formation, now it has a function, it can do something, all right? And I'm able to talk about now, right? When an enzyme is a protein, when a protein is broken down, when you break those bonds, we're breaking down the structure of that protein, right? And if you have two proteins of the same structure, right, you break some bonds, it's not going to have the same structure. So now it can't do the same thing. So denaturization of proteins changes their structure, which changes their function, which means they don't work the same. Okay? And exercise influences the structure of these enzymes. One, either because the byproducts modify the enzymes in a way to make them more active, or some byproducts inhibit the enzymes and make them less active, or they literally break down the structure, so it doesn't work. Okay, so these are all factors to talk about here, but a prime example, right, interacting with the substrate, the actual enzyme changes, structure, and so on and so forth. So when we think about enzymes catalyze reactions, okay? Essentially, they lower the energy of activation. What do I mean by that, okay? Kind of think about like, what if you have like, I don't know, a bunch of kids floating around the pool, right? If you have every kid doing their own little thing, the water's moving, they're flying around, they're being, the Little Mermaid, whatever, they're flying around, not paying attention to anybody, just by chance, they might bump into each other, right? Just by chance. Well, that's kind of what's happening inside your muscles with these, or inside your cells with these different types of substrates. There are substrates that work together. They are supposed to go together. And together, they cause a reaction in the cell. But what happens is, if you have those two molecules and they bump into each other, but they don't fit just right, they might collide but not lock into place and cause a new product to occur. Okay, so what enzymes do is, enzymes make sure that when it literally takes those two substrates and puts them together and makes sure that reaction happens. So in your cells, if enzymes weren't present, maybe these two molecules will bump into each other, 1,000 times. And maybe 2 or 3 out of those 1,000 collisions, they will interact perfectly and form a new product, right? Just by chance, they happen to fit perfectly. But what enzymes do is, instead of every 2 or 3 out of 1,000 collisions, every time they collide, they make a product, right? So it lowers the energy required for that reaction to occur because it ensures the reactions happen. Why? Because the enzymes literally put those pieces together to perform the product. Does that make sense? So when we look at this kind of diagram here, we see the reactant. See how the ball is smaller and the activation energy, the wall is much higher. It requires much more activation energy, right? And requires much more initiative or a greater, more optimal cellular environment to occur for these reactants to overcome those chances to cause the reaction to occur. When you have an enzyme present, literally it makes those reactants more powerful, more enabled to have a reaction occur, right? So it's easier to get over that hump. It's easier for that reaction to process through. Does that make sense? So what are some factors that regulate enzyme activity? One is temperature and another is pH. Somebody tell me, how does temperature change during exercise? It increases. Why does temperature increase? You know it increases. Sorry?
Muscles will give off heat. Right?
Muscles give off heat. Where does the heat come from? Why do they give off heat? Take a second. You go for a light jog, right? 5 minutes, real brisk, maybe even just a brisk walk. Does your temperature increase very much? No. Now you're on the track team, the coach tells you you're doing suicides for the next half hour. How is your temperature there? Really high, right? Your core temperature increases. Let's go back to our first comment about ATP. The harder the exercise, the more ATP you use, right? So what's the difference between doing suicides for a half hour versus a 5-minute brisk walk? What's going on there?
Using a lot more ATP.
Way higher intensity, way more ATP use with the higher intensity exercise, right? So how does that relate to heat production?
Oh, it gives off energy.
What gives off the energy?
Breaking down
The what is?
ATP. Breaking down the ATP.
Breaking down the ATP, yes, right? So the more ATP you break down, the more heat's that produced, right? How does this work? Well, let's look at it like this. Oh my God, that these similar colors are not great. Okay, we'll try one. Okay, so we have adenosine, we have phosphate, phosphate, phosphate. They're bound together, okay? Adenosine triphosphate, 3, ATP. Imagine, well, you're familiar with like a coil, like a spring, right? A little bouncy spring, right? If you have a spring with no pressure, right, you're not doing anything to it, it's expanded, right? But when you push that spring together, like say I had a small spring and put it together in my fingertips, right? Is there going to be resistance there, right? When you squeeze a pressurized spring between your fingers, what you're doing is you are storing potential energy. Okay? When you release the pressure, when you release that spring, you are releasing the energy. And so imagine in between the bonds of the phosphates is like a spring, a coiled spring being pressurized, right? When you break the bond, when you break that ATP away, it's like releasing the energy, it's like releasing that spring, right? So ATP stores energy, the energy is stored in between the bonds. So when you release or break that ATP, you hydrolyze ATP, you break it down, this phosphate comes off, energy is released to do cellular work. The other rest of the energy is heat. Okay? You break down more ATP, you break another bond, you do more work, you produce more heat. Right? So we can see that the more ATP we're breaking down, the more heat we're producing, but the more work we're doing. Does that make sense? I mean, combustion engines are much more efficient than our muscle at producing work relative to heat, right? Engines get really hot, but they do a lot of work. Like comparatively, they perform more work compared to the heat they produce versus our muscles. We produce more heat than what we do with cellular work, okay? But we're very aerobically efficient, okay? So relative to, so if we think about this, this together was ATP, right? If we just have these two, what's that? ADP, okay? And then if we just have these two, what is that? AMP. Right, so lots of energy, we're starting to break down energy, now we're breaking down even more energy. Realistically in the cell, when it's contracting, you have ATP, you're breaking down, okay, now you've got some ADP, plus some inorganic phosphates, you break down some more, now you got some AMP, then you got some more inorganic phosphates. You can still have some more ATP around because there's still energy left, but the more ATP you break down, the more ADP there is, the more phosphates go up, and so on and so forth, right? So the more ATP you break down, the more inorganic phosphates you produce, because where did they come from? Well, they came off of the ATP you just broke them off of. And if you broke off the phosphate, now it's not in ATP anymore, now it's in ADP. You break that off, now it's in AMP. Okay? So, very key thing when we start here and thinking about metabolism, a lot of these bioenergetic pathways, they all respond and are stimulated by byproducts of ATP breakdown. Okay? If you can remember that, you'll be pretty golden for all these pathways, right? All these situations where there's a lot of energy in the cell, a lot of ATP, a lot of glycogen, that's not a stimulus for increasing these pathways, right? Because they don't need to. There already is a lot of ATP. So hopefully you guys remember the bioenergetic stuff from 346. You guys remember your rate-limiting enzymes for the most part? Right? Remember which pathways we use for which type of exercise? Kind of, sort of? Okay. After today, make sure you go back and study some more of that too. The next 3 weeks are just on that. So, getting back to it. When we break those bonds between ATP, the phosphates ATP, heat's released. That increases temperature. Temperature is something that changes enzyme activity. Why? Because temperature can denature proteins, right? If temperature increases, the bonds between certain atoms, certain molecules on these proteins break and they change. So if the bonds break or change, the structure changes, which means the function of that enzyme changes. Bless you. Same thing with pH. How does pH change with exercise typically?
Decrease.
Yes.
Increase like that.
What increases in hydrogen, right? But which way is which? I heard people, I stopped because some people were saying acidity increases, and then some people were like, oh, pH goes down, right? But like, tell me the correct order, because on the exam, you could, if I gave you an option that says, oh, the acidity of the cell increased, which of these pHs is most likely to be representative of that, right? If you put the pH goes up, you're wrong. The cell's more basic, right? So there's an inverse relationship. The lower the pH, the more acidic. The higher the pH, the less acidic, or the more basic, the more alkaline. Okay? So don't get those confused, because on the exam, I do use pH or the term acidity interchangeably depending on the question. Okay? So you got to know those terms. But yes, pH also influences enzyme activity. During exercise, typically pH will go down, particularly during high intensity exercise. And we said pH goes down because of more hydrogen ions. Okay? When we, like in general, during high intensity exercise, when we produce more hydrogen ions, right? Where are those hydrogen ions usually supposed to go? To the electron transport chain, right? What actually takes those hydrogen ions to the electron transport chain? Electron carriers. Where are they? Well, they go to the mitochondria, right? But what are those two chaperone molecules? NAD8 or NAD plus and FAD, right? NAD and FAD, they latch onto those hydrogen ions. They also, carry the electron as well. They go, and now they become NADH and FADH. They go to the electron transport chain, and they drop off those hydrogens into the mitochondria, into the membrane, stuffed into the membrane space. Then they are forced down, accepted through oxygen, through ATP synthase, joined with oxygen, and we make ATP and water, right? So the product of oxidative phosphorylation is ATP and water. Why? Because the hydrogens are most strongly attracted to the oxygen in the mitochondria. So if there's oxygen, the hydrogen go there to form water and ATP. But if there's no oxygen, the hydrogens can't go into membrane space, right? Imagine like, you ever know like the subway, it's like peak travel hour, people are trying to get home from work, like you see people like push into a squeeze into a subway. I've been there, right? You can't push even more people into an already packed subway train car, right? People have to wait until it's empty or there's room to get in. The chaperone molecules dropping off those hydrogens kind of the same way, right? If it's already saturated inside that intermembrane space, you can't dump more hydrogens in there. So instead, you have these NADH and FADH kind of holding on to the hydrogens, right? And that means when metabolism's still going, if NADH are already occupying the hydrogens, when more hydrogens are produced, the cell just becomes more acidic, right? Because now all these hydrogens continue to build up. And so pH is going down, hydrogen going up. The fact that pH goes down, those hydrogens, the environment being more acidic, changes the hydrogen bonds in between molecules of those enzymes. So if the hydrogen bonds break, those bonds break, changes the structure, changes the function of those enzymes. Again, right? So here we just gave a few examples about how temperature and pH changes with exercise influence enzyme function, right? And those enzymes, the purpose of them are to mediate this exercise stress, right? Respond to the exercise demands. So, to reiterate, when we think about substrates and enzymes, as I mentioned earlier, these enzymes, they fit with specific substrates. Notice how these substrates can fit together. Remember I said, oh, maybe they'll collide and maybe they'll fit together and maybe they won't. When you have an enzyme that fits them both perfectly, you make sure that they interact and join correctly, and you always produce the product, right? So with the presence of the enzyme, you ensure that that reaction moves forward. That's what we mean by it lowers the activation energy, because instead of these happening to interact or happening to collide every so often, and maybe they fit together, when they join with an enzyme, they always produce the product. Right? So it's less wasted energy, less chance. Okay? So these enzymes facilitate, they catalyze reactions. And this is important because during exercise, we don't want to depend on chance whether we can access that glycogen if we need to run away from a bear, versus, oh, now I'm stuck, I stick to fat. You can make a lot of ATP with fat, but not very quickly. And if I need to run for my life, I need the glycogen now. I don't want to depend on chance whether this enzyme is going to turn on for me to use that glycogen. I need to make sure I can get it. Okay? So these enzymes make sure we can mobilize those substrates, or shuttle those hydrogens, take hydrolyze things, oxidize things, shuttle them away, things like that. Questions so far? All right, so we'll talk about some classifications of enzymes, right? You don't have to memorize these, but we will see that, you know, what's in the enzyme name can tell you what its function is, okay? So we have like octoreductases, they're involved in oxidation reduction reactions. We have transferases, they transfer molecules on atoms. We have hydrolases, they're involved in hydrolysis or splitting molecules. We have lyases, which they remove atoms. Isomerases, they rearrange things. Ligases, they use ATP to combine molecules, okay? Some examples: oxidases oxidize things, reductases reduce things, transferases. You have transaminases, they transfer amino acid groups. Kinases, see, point from the study guide. What are kinases? What do they do? Kinases, they transfer phosphate groups. Okay? We talk about kinases all semester. One of the ways that enzymes turn on, or proteins turn on, is they get phosphorylated. Okay? So kind of think of it as like some proteins, when you add a phosphate, they get turned on. Some proteins, when you take the phosphate off of them, then they turn on, right? Now, depending whether how many sites are phosphorylated or whether there's some other molecules interacting, maybe it's kind of like, instead of an on and off switch, maybe it's like a dimmer where you're kind of turning it on or kind of turning it off, right? You can slightly change that true activity of the light in the entire room based on the activating factors, the inhibitory factors, and the phosphorylation that's happening. Okay, so this is how our muscles will regulate specifically the demands it's receiving from exercise and what's needed to sustain or challenge that exercise session. Okay, but kinases we talk about all semester, so you got to know kinases. That's on your exam for sure. You have hydrolases, which include and dehydrogenases. Okay, some examples being proteases. They break down peptide bonds. Peptides are proteins, so proteases break down proteins. We have lipases. Lipases was on that study guide as well, right? What are lipases? Lipases hydrolyze lipid ester bonds. That's a fancy way of saying it increases. What we're involved in. Fat. What does it do to fat, you think? Break down fats, right? It's involved in lipolysis, right? So you kind of see the commonalities between, we already know about the process of lipolysis. What is the process of lipolysis? What is that? By definition, lipolysis is breaking down fat, right? We already know that. So we can see the similarity in the word lipolysis and the word lipase. What does it do? Things that ends in aces, it tells you that it's an enzyme. So this is an enzyme involved in a fat. But it does something with fat. What does it do? It breaks down lipids. It breaks down fat. So therefore, it's an enzyme that regulates lipolysis. Okay? We'll talk about lipases, specifically the lipase that regulates fat metabolism in response to epinephrine. We'll talk about that in a few lectures. Okay? So what are some factors that alter enzyme activity? Well, we already mentioned temperature, we already mentioned pH. We've also got to consider the enzyme concentration and the substrate concentration. So for example, if you have a lot of enzymes and only a little bit of substrate, there's not really going to be that much reactivity happening because there's not that many substrates for each enzyme to interact with, right? Conversely, if there's a lot of substrate and a little bit of enzyme, then that process is also going to be rate limiting because you can only have each product produced depending on how available that singular enzyme is available, right? So the best option would be to have lots of enzymes and lots of substrates so you can have lots of products being produced, okay? How do we relate this to exercise and training? Well, who's heard of that enzyme called citrate synthase? Who's heard of that before? I hope you have. It was in 346. Where is that enzyme located? The Krebs cycle, right? Actually, it's the first enzyme in the Krebs cycle. It starts off that first molecule, which is not acetyl-CoA. It uses acetyl-CoA. What's the first molecule in the Krebs cycle that's produced? What's the enzyme name? I just mentioned.
Oh,
Citrate synthase. So what's the product? Citrate, right? right? So it's the first enzyme that produces citrate, okay? So citrate synthase is an enzyme that is at the start of the Krebs cycle, which is it an anaerobic or aerobic pathway? Aerobic, right? Because the process of the Krebs cycle produces carbon dioxide, completely oxidizes the carbohydrates, produces an acetyl-CoAs, right? And then those hydrogens go to the electron transport chain to produce ATP. They don't directly produce ATP, but it does completely oxidize those substrates. Okay, so yes, the Krebs cycle is an oxidative pathway. So we see with aerobic exercise training, chronically, aerobically trained individuals have more citrate synthase in their muscles. They have more of this enzyme that makes aerobic exercise metabolism more effective, more efficient. So do we see how if we have more of that aerobic enzyme, we can actually use more substrate aerobically just by having a higher concentration of that enzyme? We'll also learn aerobic training lets us hold more glycogen. It lets us hold more fats and lipids in our muscles. More enzymes, more substrates, more products, more ATP. Does this make sense? That's actually adaptation right there. Now we've also got to consider, yeah, you have more enzymes, or maybe you have more substrates, but there are other factors that specifically activate enzymes or inhibit enzymes, and we'll get into those in some of these next few slides on glycolysis. Okay, so yes, there are these rate limiting steps, but if there's byproducts of breakdown like AMP or ADP or even ATP, those can interfere with the enzyme from working to either keep the enzyme off or turn the enzyme on, even if there is more stimulating factors or not. Right? So we'll get into that a little later. Are we following here? Do you need me to repeat anything? So, as I mentioned before, temperature and pH both influence enzyme activity. A small rise in body temperature can increase the activity of these enzymes, right? And the increase in temperature makes the enzymes more active. That facilitates bioenergetics to start speeding up. But at some point, if the temperature is too high, it denatures the protein. So then the enzymes do not work effectively anymore. Same thing with pH. When pH decreases too much, when pH decreases or slightly increases, it can make those enzymes work more effectively. When it's too drastic, the enzymes do not function anymore. Okay. Now, realistically, in humans, are you going to see in your muscle pH get down to 2? No, there hasn't been any recorded, in humans of muscle acidosis of the pH that low. But if we're thinking about physiological range, yeah, you could get down to like 5, maybe 6, probably low 6s, maybe 5, right, depending on the intensity of the program. But these enzymes work in a physiological range, right? So optimally, we have a pretty good range right here: higher pH, lower pH, or higher temperature, lower temperature, right? But if it gets too far outside of that range, then the reaction activity or the reaction capability of those enzymes goes down. Same thing with pH here. Obviously a much more narrow range, but you can see even between 5 and what this is a little over 9, maybe 10, right? Much more narrow range. But pH in human muscle is not really going to change much further than 6, right, on the low end, okay? But just those small changes can influence enzyme activity pretty drastically. Cool? Okay, so definitely on your exam, you gotta know lipases and kinases for sure. We talk about the temperature and pH on the exam as well, so make sure you understand those concepts because they are coming for you. All right, so we'll switch gears a little bit and talk about the fuels for exercise. We think about carbohydrates. They are monosaccharides, disaccharides, and polysaccharides. We have glucose, which is often comes from blood sugar, right? We have the stored form of glucose, which is known as glycogen. Okay, glycogen is usually stored in our muscles and in our liver, and it's synthesized by an enzyme called, glycogen synthase, right? So the function of this enzyme is to synthesize glycogen, it's in the name. Okay, Glycogenolysis is the breakdown of glycogen into glucose. Now I'm very much a visual learner, and I always thought, like, okay, glycogen is just this word you see on these PowerPoints, right? It's glycogen, it's a word in textbook. Glycogen, it's a bar graph, right? Oh, what the heck does glycogen even look like? What is it? Glycogen are actually granules. It's like a spherical ball, okay? So if you look on this electron micrograph, all these little teeny tiny black dots, those are glycogen granules. Notice how they are all interspaced within the muscle fiber, right? Here's the sarcomeres, right? Z disc, M line, they're kind of there, right? Notice how they're all interspaced all throughout the muscle fiber. Now, what do these granules look like? They are spherical. We have at the center of that granule is something, a protein called glycogenin, that is essentially just the starting block of this glycogen granule. And then you have a bunch of glucose units linked together and branched off of each other into this spherical unit, okay? So essentially it's kind of like you look at a tree trunk and then all the branches that branch off of that trunk. It's kind of how we look at this, this glycogen granule. Instead of just a tree, it's a sphere, okay? And so when we talk about glycogen breakdown, all we are really looking at is breaking off each of these glucose units off of this larger glycogen granule. Okay, so glycogen is the stored form of glucose. Why? Because it's a bunch of glucose molecules branched together. Glycogen breakdown is the breakdown of glycogen into individual glucose units, which are then used in metabolism. Okay? Does that make sense? Cool. Now, as I mentioned before, if you're untrained, you usually have around this much glycogen inside your muscles. If you're trained, your muscles are able to store more glycogen in them. If you're very well trained, you can hold even more. This relates back to, we just talked about enzymes and substrates. Very, very well trained people have very high concentrations of these enzymes that metabolize